Hello and welcome to Chapter 17, which is nucleic acids and protein synthesis. We're going to be getting into some real deep biochemistry here, and in addition to the formal lecture which I plan on delivering on Monday, I thought it might be a good idea to do some videos on this as a sort of a supplement, because this is a lot of material. This video is going to take up the first two sections, 17.1 and 17.2, which are going to be the components of nucleic acids first, and then after that a little bit about the primary structure of nucleic acids. So we're on 17.1 right now, and as always, your text has this little clinical update type of thing. And the point here is that nucleic acids, which carry the genetic material of the organism, which could be a one-celled organism or a virus for that matter, and all the way up to us humans, the nucleic acids, at least the DNA, carries the instructions for making the organism, and on top of that, DNA also has other regulatory functions. So it controls a lot of things that go on in the body. And particularly with nucleic acids, and particularly with DNA, changes in the DNA, disruptions in the DNA, damage to the DNA, mutations and so forth, can cause major problems. You have genetic diseases, we've already talked about that here and there, but it can also cause cancer. Now, the aspects of nucleic acid chemistry that deal with that are beyond the scope of the course, but there's a lot of things programmed into the body by means of the DNA well, DNA it's a big actor. It's very important. So, and as I said, cancers do have a genetic component. It's unrestricted cell division, unstoppable. You have a situation where a cell which is theoretically programmed to die after a certain number of replications, divisions, doesn't do it and it just keeps growing. And that's very bad, with some very negative repercussions for the organism. So we're going to get started here in talking about the structures of DNA, of nucleic acids. There's two kinds. There's DNA and RNA, and you're going to know more of the details as we go through here. General structure of the building block of a nucleic acid is going to be a nucleotide. A nucleotide is going to consist of a nitrogen-containing base, which you see there in blue, a sugar, which is either going to be ribose or deoxyribose. They don't have all the hydroxyl groups in the diagram there, which is sort of yellow, and a phosphate. In DNA, deoxyribonucleic acid, which is going to be ribose without a hydroxyl group here at this 2 carbon. You're going to see more complete pictures of these things later on. DNA, there's 4 bases: adenine, guanine, cytosine, and thymine. Those are the names of these things here. RNA uses the same bases, with the exception of thymine being replaced by uracil. So if this is DNA and we have a thymine, if the equivalent RNA base is going to have uracil in it. We'll see that as we go along too. The bases are derived from 2 basic structures, 2 basic ring structures. One is pyrimidine, and you see the formula or the structure of pyrimidine, the molecule pyrimidine, which is an organic molecule. And there are also purines, and you see the structure of purine as well. The nitrogen On the purine, there's nitrogen on the, with this one here, which has a hydrogen on it. Then there are other hydrogens on the molecule, not on nitrogen. If you add up the number of bonds, you know that nitrogen can form 3 bonds, carbon 4, and it's like here's a carbon hydrogen bond, carbon hydrogen bond, carbon hydrogen bond, and the same goes for these 4 carbons, which make the angles on this without the nitrogens on the pyrimidine ring. The purine bases in DNA are adenine and guanine, which is the same as an RNA, and the pyrimidine bases with the single rings are cytosine and thymine. We'll see the structures momentarily. And again, RNA does not use thymine, it uses uracil. And here is the summary of all the bases used in RNA and DNA. Your RNA and your DNA is made of, among other things, I mentioned the sugars and the phosphate, but these are pretty much the building blocks. Pretty amazing for such complicated molecules as you're going to see nucleic acids happen to be. And we'll go through them. Starting with cytosine, we're looking at the pyrimidines first. It's abbreviated capital C, and it shows up in DNA and RNA. There's uracil. Oh, let me go back. The cytosine, you have a carbonyl on the carbon, the one carbon between the 2 nitrogens, as you see, and then moving around the position. Well, I'm going to give you the position numbers here. The cytosine, this is position 1, and going around, it's we were at position 4 where you have an amino group on cytosine. At position 2, there's a carbonyl carbon. Uracil is actually fairly similar. The difference between cytosine and uracil is at position 4, at this position where I'm hovering over here, there's a carbonyl compared to an amino group here. And then there's, of course, thymine, which is almost like uracil. It has a methyl group over on the 5 position here, as you can see. Don't worry too much about the numbering. It gets a little complicated. Now, that's the pyrimidine bases. In DNA, you have cytosine and you have thymine. In RNA, you have cytosine and you have uracil. Those are the bases that are used in the nucleotides, and we'll go on. We'll be building these things up as we go along. On the pyrimidine bases, adenine is the simplest. You see the amino group is over on the 6-membered ring, all the way over on the other side from where this hydrogen is here. And this position on the pyrimidines, this position on the purines. So this position is on the purine ring, where I'm circling here. This is actually the 9 position, and where this amino group is here is actually the 6th position. I'm not going to go into the numbering. It's probably better that you remember you draw the picture and you know pretty much know. You draw it with this orientation, with the 5-membered ring to the left, the nitrogen that contains the hydrogen, the bond to hydrogen on the bottom left here, and then the other. You just see where the orientation of the amino group here. You see it's up near the top, and then on the guanine you have an amino group between the two. These are the 1 and 3 positions, but it's the amino group here and the carbonyl group. It's a little bit difficult without the numbers, but the numbering systems are quite a bit different between these two, and not too many books actually use it beyond showing it in the first time they show the structure of these things. So, but these are the structures of the bases. Moving on, the next component is a pentose sugar. Now you remember trioses, tetroses, pentoses, hexoses, and even heptoses. Well, the pentose sugars are 5-carbon sugars. They're aldopentoses, which if you go back to your textbook and look at the chapter on carbohydrates, you'll see that you've actually seen ribose. And then they have deoxyribose in there, but deoxyribose is a ribose molecule with no oxygen atom on the 2 position. So let me blow this up, show you a little bigger. If I can find a place to zoom. View. There we go. I'll just blow this up to 100% so you can see a little better. This is the way we draw ribose. Ribose exists as a 5-membered ring, and we know you already know that these sugars, the 5 and 6 carbon sugars, do form rings like this. And the ribose differs from deoxyribose only in the 2' hydroxyl. So you see in the deoxyribose that there's no hydroxyl on the 2' position. And the reason we call this the 1', 2', 3', 4', 5' is that technically the bases have numbers too. They are the unprimed numbers if we were to write out the whole structure of these things. But I don't believe you're going to really see that, and you're going to have to worry about that in your text. But for historical reasons, let's say you want to know that these are the 1', 2', 3', 5', and so forth. And that's it. I mean, those are the sugars used in nucleic acids. There's ribose in RNA for ribonucleic acid, and deoxyribose in DNA for deoxyribonucleic acid. So the D means it's dehydroxylated, unhydroxylated at the 2' position. That's all that means. But it has profound implications for the actual properties of the molecules. So we have the first order of structure. I talked about nucleotides, but there is a lower order of structure that is above the individual components, the sugar and the base, is the nucleoside, which is a combination of a sugar and a base. And it is produced when the nitrogen atom in a pyrimidine or a purine base forms an N-glycosidic bond to carbon 1, C1', of a sugar, either deoxyribose or ribose. And to get the name, you take the base, change the ending to ocene for purines, and idine for pyrimidines. So we'll see how that works here. Let me zoom back. Here's the adenine, and let's go back to here. You see ocene for purines, and remember, a purine is the fused 2 rings, a 5 and a 6-membered ring fused. So this is going to be adenosine. As you see, they write adenosine here. And if this sugar were deoxyribose, it would be deoxyadenosine. Simple. And you remember there was the glycosidic bonds in the carbohydrates linking sugar molecules or monosaccharides together to form chains. Well, there are glycosidic bonds with an N, with a nitrogen, where nitrogen is in there. And here we have beta N glycosidic bond labeled here. So you see that it is very analogous to, let's say, the glycosidic bond in sucrose, lactose, maltose, things like that. And notice they have also here a description of what exactly happens when the adenine and the ribose condense to form the adenosine under the control of an enzyme, of course. You're essentially removing these two hydrogen and a hydroxyl to form a water molecule, and I have these things labeled on this slide, so you see that you're keeping all the atoms there, conservation of mass and all, and that's how this thing is going to be formed when the enzymes work on it. Nucleotides are the last step here until we start polymerizing these things, and nucleotides are nucleosides with a 5' phosphate. What's a 5' phosphate? Well, we're going to go through this first. You're going to see that when a phosphate group reacts with the group on carbon 5, the 5' carbon of either ribose or deoxyribose to produce a phosphate ester, we only have the only nucleotides are 5' monophosphates. So those are the only 5' monophosphate nucleotides found in RNA and DNA. There are other structures, there are other compounds which you will see named as nucleotides. You're going to see it in the next chapter when we get into metabolism, but we'll get to that. They don't occur in nucleic acids. These are the only ones that occur in nucleic acids. So here we have the nucleotides, a complete nucleotide, so you can see them. Let me blow this up a bit. You see the structure in each. Here's adenosine monophosphate, guanosine monophosphate, and we'll move up a bit here. And you have the cytidine monophosphate, uridine monophosphate. Notice the adenosine is the sugar base. Guanosine is the sugar base combination. Cytidine is the sugar base combination, the nucleoside. Uridine is the nucleoside here. Deoxythymidine it's always going to be deoxythymidine, remember, for the nucleoside. That's the nucleoside. And when you put this 5' phosphate onto the molecule, you now have a nucleotide. So it is now adenosine 5' monophosphate, guanosine 5' monophosphate, cytidine 5' monophosphate, uridine 5' monophosphate, and deoxythymidine 5' monophosphate. And now uridine, UMP and DTMP are one's in RNA, one's in DNA, whereas you have the adenosine and deoxyadenosine, guanosine, deoxyadenosine, cytidine, deoxycytidine are going to show up in both. Okay, we have the names. I think you've already picked up an idea of what the names are like. The DNA nucleosides add deoxy to the beginning of their names, and then the RNA and DNA nucleotides are the nucleotides. Nucleosides add just deoxy beginning, added to their names. The nucleotides are simply the name of the nucleoside with an added 5' monophosphate at the end. So if you're looking here, there's adenosine or deoxyadenosine is the nucleoside. The name of the nucleotide is simply adenosine 5' monophosphate, deoxyadenosine 5' monophosphate. We use the A, G, C, U, and T to represent the bases, and we often just use these when we're writing in the whole molecule. A lot of times we just write the string of the A, G, C, U, and T, because in RNA and DNA, depending in either one, the sugar and the phosphate is going to be there either way, so we just write the sequence of the bases. That's the nucleotides only differ in the identity of the base. So we have the nucleosides and nucleotides in these things, and we see there's the base, the nucleoside, and the nucleotide. It's a fairly logical progression from the adenine, and then you go to adenosine, or in DNA, deoxyadenosine or adenosine in the RNA nucleoside, and then monophosphate. You see here they don't write the 5' monophosphate. That's understood, but it is at the 5' position. So they're how they're written. So this is a nice way to put down and organize the names of the bases, the nucleosides, and the nucleotides, and that's really the structure of these things. That's their chemistry is. Nucleotides have a very complicated chemistry. They're more than just, in many cases, they're more than just a component of a nucleic acid. And again, we're going to see that when we get into metabolism next week. Another summary is table 7.1 here. The components of the DNA and RNA. There's a base, there's a sugar. Bases and sugars combine to form a nucleoside. Nucleoside combines with a base and phosphate to give you a nucleotide, and I see that's a little bit of a mistake here on the slide. Let me change this. Fix that one. I've been going through these slides pretty closely this week, today actually, and I have made quite a few changes, but this one got by. But there's your nucleotide, and then they form nucleic acids. They polymerize. They form long chains of nucleotides to form nucleic acids. We're going to do that in the next section. We're going to talk a little bit about that. Okay, so learning check. Look at this here. Identify the following bases as a purine or a pyrimidine. One of the ways that I keep these straight, well, the only way I really have to keep this straight is to realize that the simple, well, if you want to call it a one ring system versus a two ring system, the one ring system being simpler. Well, the simpler ring system has the more complicated name. So a cytosine is a pyrimidine and adenine is a purine. By the way, the carbon structure, or this ring structure of adenine, of purine, is actually the same ring structure in caffeine. So purines are quite, they show up in a lot of different ways in the body as metabolites or intermediates in the synthesis of these things too. So adenine is a purine, cytosine's a pyrimidine, as you see here. Learning check 2. Identify each of the following bases as a component of DNA. Now remember, it's uracil in RNA, thymine in DNA. Remember that. Okay, so guanine's going to be both, adenine's going to be both, thymine's going to be DNA, and uracil is going to be in RNA. And that is how this works. Okay, I'm going to stop at this one. I'm going to do these sections standalone. So the individual videos are going to be shorter, but I'm going to try to get all this in before Monday. And if I don't get it in before Monday, at least I'll have it done, and hopefully you'll have a chance to look at this, particularly the beginning sections. And I think my substitute covered a little bit of this. I don't know how much. I haven't asked her how much she did, but I mentioned she should try getting started on nucleic acid. So you've probably already seen this, and here it is again. Hopefully this will be a useful reference, and we'll go on to section 2 next.